With the choice of H5N1 for our research, we really tried to hit two birds with one stone. On one hand, we tried to address the very fundamental question of how influenza viruses become airborne. By using H5N1 as the model virus, we would directly assess the risk of H5N1 becoming airborne. We know particular genetic changes that have occurred during the pandemics of 1918, 57 and 1968. And we use that knowledge to genetically modify H5N1 bird flu virus and try to see if it would also acquire this ability to become airborne. And initially that was not the case. But we then did an experiment of natural selection in the ferret model by serially passaging the mutant virus in ferrets, in the nose of ferrets. And after 10 passages in ferrets, the virus had become airborne, so it could be transmitted by coughing and sneezing and breathing between ferrets. We also characterized the viruses that were transmitted by the aerosol route to identify the mutations that were associated with airborne spread. And that it was actually fairly simple for that virus to acquire that ability. It only took in between 5 and 10 substitutions in a few genes to become airborne. Genetic changes that occurred in the virus were quite similar with previous pandemic viruses. And we further investigated whether this virus would be sensitive to antiviral drugs. By knowing exactly which mutations or which biological traits we have to be on the lookout for during outbreaks, we might be able to eradicate those viruses specifically that are a direct threat to public and animal health. We can better assess the effectiveness of antiviral drugs and vaccines before the viruses actually emerge in the field. Biosafety and biosecurity issues around this manuscript are really minimal. We perform our work in a very safe laboratory. The facility is highly secured also and is under inspection by two governments, the Dutch government and the US government. You need very trained specialist virologists to do this type of work, and so it's unlikely that any lone wolf would repeat this type of work. On top of that, we show in this manuscript that the virus is not particularly lethal once it's transmitted via the aerosol route. In ferret, none of the ferrets die, and it's not particularly efficient in transmission, so it would be unlikely that if this virus would be released from the laboratory, it would actually cause a massive outbreak in humans. Well, the future of this research is really to increase our understanding about the specific mutations that we identified, the biological traits that are associated with those mutations, so that in the future we really can predict which viruses in the field are more likely to become airborne at any given point in time. Increasing our fundamental understanding of the genetic changes that are occurring in viruses once they become airborne. And also research on how generic our findings are to other influenza viruses. But these are really fundamental projects for which much more research work is needed.